Hello, everybody. I'm Carl Alston from the Leukemia Research Foundation. Welcome to our Blood Cancer Awareness Month live stream with Dr. Andres Blanco, who is an assistant professor of medical or biomedical sciences at the University of Pennsylvania School of Veterinary Medicine. Thank you so much for joining us, Dr. Blanco. Really appreciate it.
Thanks a lot for having me here. I'm really looking forward to talking with you.
Well, great, because my first question is coming now, although I should remind everybody that if you would like to ask questions during this time, all you have to do is write your question in the comments section of Facebook or YouTube, and that question will come to me, and then I will ask it, ask it of Dr. Blanco. So, continuing, Dr. Blanco received a research grant from the Leukemia Research Foundation a little bit over a year ago. And the title of that project was Dual Targeting of LSD1 and KAT6A to Induce Therapeutic Differentiation in AML. So, Dr. Blanco, can you tell us, number one, what the heck does that mean? And number two, what did you learn from the project? Because that was a little over a year ago that we funded that.
Sure, yeah, I'd be happy to. Let me start with what the heck does that mean? Kind of a clunky title there, maybe. So my research, Carl, is on the interface between basic cellular biology and studying how cells grow in the leukemic context. Onto the interface of that and more applied therapeutics. And that's because what I'm really interested in is a therapeutic approach for AML, acute myeloid leukemia, that's a little bit different from chemotherapy, and it's inherently low toxicity. It's called differentiation therapy. And the idea, the goal of this approach is to give cells treatments that make cells, rather than just giving them high toxic chemos to kill them directly, it aims to make cells essentially stop growing and then just die of their own accord, a more natural cellular death. And this has been curative in one subtype of leukemia, but has not worked in the other subtypes. And I'm trying to figure out why. And where this project, what it focuses on is that there's one gene, one target, a gene called LSD1, which is in the title, that is very important for regulating how leukemia cells grow. So much so that there are inhibitors against this gene that are in the clinic right now, which is really exciting. And what we found from the initial clinical trials and from my own research is that targeting of this gene LSD1 alone it's very promising, but it may not be sufficient to completely stop the growth of leukemia cells. And what I found in my research is that there are other genes that work in concert with LSD1 to regulate cellular programs controlling cell growth. And one of them is a gene called KAT6A. And I found that if I inhibit both LSD1 and KAT6A at the same time, I get a much more powerful response. And so that's tremendously exciting to me. And this project, the goal is to figure out how these two genes are working together and what are the best strategies for targeting them, inhibiting them at the same time. So yeah, that's what the project is about. And the second question, Carl, was what did we learn from it? Is that right?
Yeah, that's right. But basically, I just wanted to say, so, the whole point is to try to get to a point where, at least with certain forms of leukemia, you can treat it without using chemotherapy.
Right. That's exactly the idea.
That helps patients a lot because you don't have to deal with the fallout from doing, having chemo and all of the illness that might bring along, or the discomfort that it might bring along, depending on, which form that you have. So, okay.
That's exactly right.
And then, so what did you learn from the, from the research, and kind of what was it, what you expected, or something different, or what?
Yeah, we learned a lot of things from it. It's been a fantastic project. I've just been thrilled to be able to do it with the help of the Leukemia Research Foundation, and we learned a lot. We learned In particular, these two genes, LSD1 and KAT6A, they regulate the properties of growth of the cell in kind of independent ways. And what that tells us is that we seem to get such a good response with leukemia from leukemia cells when we target, inhibit both these genes at the same time, in part because we think that we need to target both of these cellular programs at the same time. So one thing that tells us is that first and foremost, this may be a combination therapy that we could see with further work, may have the potential to go further and be of consideration, in clinical trials in and of itself. There's a lot of steps we have to go through to get there. That's one possibility, but we also learned about, really what are the programs in the cell controlling cellular growth that each of these genes regulate. And so from that, we found that in addition to KAT6A and LSD1, there are other genes involved in these programs. And so there may be other combinations we could try depending on targeting other genes in combination, depending on what inhibitors are available or can be designed. And but just to underscore what you said, Carl, indeed, the idea is that chemotherapies, they have been very, very effective in ways for a long time in leukemia, but leukemia treatment still relies primarily on chemotherapeutics when there are more targeted, gentler therapeutics out there that, in particular in AML, whose patients are oftentimes of a bit more advanced age and may not tolerate chemotherapies as well. This is all the more so an important therapeutic approach. And there, as I said, in a different subtype of leukemia, it's been curative. So there's a precedent for the cure. We just want to figure out how to get it to work, in a more broad spectrum of leukemias.
Right. So that's really pretty cool because now it's sort of, you've gotten to the point where you're using a medication that's already out there, and you're trying to see if it combined with another medication or other medications will, be effective for people. And if that is, then that's really good. That provides another treatment right off the bat, at least one, and maybe several other treatments for people. And you don't have to go down don't necessarily have to go down the chemotherapy road, which will be more comfortable for patients. And that's kind of how it fits in the whole world of blood cancer treatment. That's a new thing that will happen for the world of blood cancer treatment, right?
Yes, that's exactly right. That's exactly right. Yeah.
Cool. So we were pretty happy as the Leukemia Research Foundation to provide you with funding this year. We are fortunate to have some help too from the Teddy Kim Foundation in New Jersey to support that grant. So thank you very much to the Teddy Kim Foundation. So how did the funding help you with your project and also help advance your career?
Wow. Well, Carl, I'm glad you asked that because I can't overstate how helpful this funding was from the Leukemia Research Foundation and from the Teddy Kim Foundation. Can't overstate how helpful this was to the project and indeed to my career. So, Carl, I'm fairly new as a lead researcher, a professor who, I'm now running my own lab. I started up my lab up about 2.5 years ago. And so, when I got this funding, I was, a little, about 1.5 years into my starting up my lab. And in this early stage, of doing research, that's when, things are the most fragile in a way for young investigators, because really over time, as as an investigator gets more established and has more of a track record of leading his or her own lab to big successful findings, then it becomes easier to get large scale federal grants, which will support a researcher in the long run. The thing is, as a young investigator, it takes time to get there, and so in those first few years, this is like basically right now when I've had to basically be able to build up the research program within my lab, really get it to thrive, and to get myself to the point of producing the results in my program that will get me eligible for federal grant support, which will hopefully just launch my lab really into the future. But it's at this precarious time when would be very hard for me to otherwise get funding from the NIH or other federal sources. This is a time where absolutely every dollar from this grant was so helpful to the project. And specifically, how this helped is that this project is really the one that I proposed here for the Leukemia Research Foundation, and thankfully the one that they funded. This is the most exciting and the best project that I have going in my lab. I believe it has the most potential to have a real therapeutic impact on patients. For that reason, I wanted to put everything that I had into it, and I wanted to not be limited by finances, within reason, obviously. But having that extra money to go to fund specifically this project, that took everything to the next level. That allowed me to say, when we knew we've got an experiment that we've got to do, we've got an assay that we need to do, and it might be a little bit expensive, but we know it's the right thing that we need for the science. This grant let me do that. And the results have been fantastic. We've just finished up the main component of this research, this project, and the timing is remarkable. We're just actually this week in the process of submitting a manuscript for publication based on this work. We're submitting it to the top cancer research journal in the world, and we don't know if it will be accepted to that one, but I think it will land somewhere good where people can, other people from basic researchers to clinicians can read about it. And honestly, it is no stretch to say that it's this grant that made the difference.
Well, that is fantastic, and I want to share a note that I just got from Mr. Kim at the Teddy Kim Foundation, and he said, "We are excited and thankful to have been part of your research. Keep up the great work." And that's from the Teddy Kim Foundation. So congratulations and thank you. That is really cool. And it's really cool to learn that you are about to submit the results of that research for publication. I mean, that's a big deal.
Yeah, this is it's huge for the project. It's kind of from a bit of a more basic researcher's perspective. I'm a basic research guy studying applied blood cancer problems, but from a basic research perspective, this is where you put everything out to the scientific community, the scientific and medical community. It's through publications, peer-reviewed publications. Ideally in some of the top journals that are read worldwide. This is for a project, this is our kind of best case scenario, is that things come together and we publish it in a leading journal, and we're fingers crossed, we're hopefully getting there real soon. And it's exactly right. It's so funny. It is actually this week that we've been in touch with the journal, talking about whether we put in what are called pre-submission inquiries to ask, is this the type of thing that the journal might potentially be interested in? And they said yes, gave us the green light to at least submit the publication. So that's just tremendously exciting for us. And thank you, especially for the Teddy Kim Foundation checking in. That means so much to me, and thank you so much for the support.
Well, like I said, congratulations again. That is just, that's outstanding, and that's what we hope to do. We want to fund really innovative and good research. It's great to hear that your research is going to hopefully be published, and then, of course, now we hope that not only is it published, but that you end up getting more funding to go on to the next phase, whatever that is, with this research, and that, as soon as you can, just find a cure, will you? That's all we want. No pressure there.
We can.
So actually, what is the next thing that you want to do? You know, if things, you know, keep going well, what's the next phase, if you can say?
Yeah, yeah, absolutely. Yeah, well, I mean, I couldn't be more excited because with this project, I think getting going as well as it did and yielding the interesting findings that it did, it also has opened up some very, very interesting, has very, very interesting implications for understanding what we found to some extent in greater depth and to some extent more broadly in trying to understand how universal are the findings that we have in certain models of leukemia. How much are they, particular to certain subtypes of leukemia, versus, how universal might they be? And that's another way of saying that in the era of personalized cancer medicine, where, a leukemia patient is going to go to the clinic and have, in many cases, have their leukemia genome sequenced, which will give, a personalized, genome readout of exactly what are the genetic mutations that are that cause this leukemia. And patients can, based on that information, along with, all the insights that the clinicians treating the patients have, this oftentimes informs decisions about whether the patient, might respond best to this or that therapy. So, we would like to figure out for this combination therapeutic approach we are working on, if it were to go to the clinic, which patients do we think would respond best? Are there a certain subtype, patients that have, a certain, type of genetic mutation, a different type of genetic mutation? So that's one thing about kind of going a little bit more broadly and figuring out how broadly applicable these findings are. The other is going deeper and extending, as I mentioned before, we found really, and this is the thing I really like about integrating in, basic research to a real, applied cancer biology problem. We really found a lot about, when we target these two genes, why is it that it's these two together? And we have pretty nice methodology for trying to understand, try to figure out really exactly what these two genes are doing. To what extent do they work in tandem? What extent do they work separately within the cell? And what, as I alluded to earlier, what we found from that is that it's these two genes that make proteins that tend to work mostly on regulating independent programs of the cell cycle, which regulates whether cells continue to grow or not. They work independently of one another, but not each one not in isolation. Each one works with other proteins. And so we would like to know, okay, hey, guilt by association, right? Like what if we start targeting some of these other proteins that are, that are working together with either KAT6A or with LSD1. So that would be a next frontier that we want to study. But the first last thing I'll say on this is that before you can to enable you to go on and take that next step as a researcher, you've got to get established, and you have to show that carve out an area of finding that's your own, that's important, that that gives people who are backing you and funding you, gives them reason to continue to support your research. And again, as I mentioned, Carl, this really is the big project from my lab that's going to be kind of the one that we want to initially be known for, and that we want to let people know, hey, we can expand on these findings, we can make them, we can increase them, make them bigger and better. And so that's, we're by no means, stopping here. This individual study is, hoping to, wrap up and publish it. But the implications, therapeutic level, and and the implications for where my lab will be moving on to, they're rooted in the success of this project.
Well, cool. That is really exciting. So speaking of that, let's talk a little bit differently here. What drew you to science? Why did you choose science as a career?
That's an interesting question, actually. I don't have the what some people might think of the kind of science nerd, like background. I wasn't a kid who just wanted to go outside and just everything was, wanted to study life or, everything around him. As a little guy, I mainly liked, playing baseball and, being, really running
Yeah.
around and all that. And it wasn't until a little bit later that I really got into science. I mean, I always did like academics, I guess in high school and especially in college. I liked all aspects of of academics, but I really started to gravitate in my classes more strongly to kind of like math and sciences. And I thought a little bit about what I could do, as going back to, I would say this is probably in college that I really made the commitment to molecular biology, molecular and cellular biology. I thought that, well, I like science in general. If there's a science I'm going to study, this is the one where I could do the most for, to say that, hey, I did something on this earth, and the one where, the most has the most potential to help people just in terms of what I would bring to the table as a scientist. And
Right.
so it's pretty much that. And then it was in graduate school when I really started studying cancer biology.
Yeah.
And then at that point, I was really, from there, there's probably kind of no turning back, because then I really did see, hey, this is real. I mean, like indeed, if I'm a scientist, I'm trying to do, research on cancer biology. I'm trying to do it, at the highest level. And I saw that, hey, I was just a young grad student, but seeing, people around me, like other researchers, seeing, professors having findings that actually were picked up by potential, sometimes by clinicians and led to clinical trials. I thought, well, I mean, if I could do, half of that, in my career, that's what I want. So that's what
Yeah.
it is.
So you are you are definitely doing that. The work that you're doing, you know, probably already has, but certainly will have impact on patients. So that's pretty cool. You set that goal, and it looks like you're going in that direction. So that's great. So you're also a parent, right?
Yes, I'm thrilled to say I'm a relatively new parent. I have one son. He's 17 months old, and
Yeah.
he's fantastic.
Well, that's pretty awesome. How do you make this work with having this important work that you're doing and achieving some kind of balance so that your son will know what you look like, who you are, that kind of thing. But let alone your wife, she probably wants to know who you are and stuff too, because she has a son to take care of, and you got to be around.
Well, Carl, it kind of depends what day you catch me on. Sometimes I'd say, well, the solution is I'll sleep when I'm dead. but
Yeah.
more, more seriously, it presents a challenge to my schedule and logistically. But I would say that, I like to think I can meet that challenge just with it's kind of the inspiration, the driving force. That just feel more meaning every day, and when I come home, science is like anything, it's hard, and there are successes and there are failures, and I guess it's probably like this with anyone when they come home from, whether it's work or it's a hobby or it's something that they do, and if they don't have the greatest day, for me and probably for most parents, you see that little guy or girl, and the challenges that you had, in your work or whatever it might be, they recede a little bit, the kind of the feelings about them, and or the kind of any sort of feeling of being discouraged recedes, and I feel the next day I feel fresh and motivated again. So I think that having the kind of inspiration it brings kind of I would say mitigates the challenge that it presents to my schedule. And my wife is a scientist also. She's also in biology. She actually studies ecology, so not biomedical sciences. She's a professor also. So
Wow.
we just we do our best with it. And I guess all I can say is that, to put it real simply, I was type of guy that I wasn't sure, if I didn't know if really, having kids was the thing for me. I feel really kind of dedicated to the lab, and all that. And well, my wife talked me into it, and all I can say is that I'm so glad we did, and she is too. So I guess that's kind of the proof is in the pudding.
Well, congratulations, you guys. You're doing great. I know you're going to do great. You're going to do great in the lab, and I think you're going to be a great dad too. So just keep up both things at the high level that you're already doing. So I have one more question. If it wasn't science, what would you have done?
Oh, that's
a
great question. Yeah. I guess it means, what would I have done in fantasy land, or what would I have done in reality? Fantasy land, Carl, I would have been a Major League Baseball player.
Okay.
That's what I originally wanted to do. Of course, reality set in, and I realized that wasn't the path. Joking aside, actually, I would have to say, if I didn't do science, one thing I've always actually been interested in is science and more health policy, especially as pertains to in the global context. So like global health policy. And I got interested in this actually because grinding it out in the lab, long hours, in graduate school, doing my PhD and all that, I just got to wondering, like how much will whatever I find, and what does it have the impact to do, and what would be the limitations to, even if I, do my part, so to speak, by doing, effective scientific research on biomedical problems. Does that guarantee that those biomedical problems are going to be, it's going to help solve them? And in maybe in some parts, yes, but I just got very interested in how there's an enormous level of blockade, between science and medicine, and then getting these, amazing findings and amazing, treatments, getting them to patients. That's really, it's not a scientific block. It's more of a policy-based block. All the more so with developing world, like infectious disease, for example. And so, I really got quite interested in organizations like Doctors Without Borders, the World Health Organization, things like that. I think if I had kind of called it quits on the lab life, I think that probably something in that direction is maybe what I would have tried to do anyway.
Well, then, so the real deal is, no matter what, you were going to have an impact on the world with whatever you did, whether it's going to be this way or the other way. So that's cool.
We all have our way to try to make our impact. Some of us do it through our work, some of us do it through things in our personal lives. So, science is my thing, and so just try to do my best with it.
Cool. Wow. Thank you so much, Dr. Blanco, for participating in this stream. I thank you so much for doing it, and I thank everybody who has been watching this stream over time. Wanted to let you all know that it is Blood Cancer Awareness Month, and there will be a few more of these streams. So pay attention to our Facebook and YouTube channels to see what's going to be happening next. We'll have a stream going on tomorrow morning with a young AML patient survivor. We'll be talking to her at 11 a.m. Central Time. Also, there is a patient education program coming up. The first one happens on Wednesday, September 16th from 7:30 to 9 p.m. And on that day, we are going to be talking about CLL and small lymphocytic lymphoma. And then 2 weeks later on a Wednesday, that would be September 30th, again from 7:30 to 9:30, we're going to be talking about aggressive non-Hodgkin's lymphoma and CAR T-cell therapy and other immunotherapies. And then 2 weeks after that, on October 14th, we're going to be talking about acute myeloid leukemia. So again, stay tuned to our social media channels to find out more about that. And if you'd like to listen in, all you have to do is register. They're free programs. All three of them are free. If you go to allbloodcancers.org and you click on the virtual education programs at the top of the page, you will see the link to all three of those programs. In fact, I think I am going to try to put the link in your comments box, so you should see that momentarily. And if you go to that link, you'll be able to see what's going on there as far as that patient education program. Also, if you just like to help us during Blood Cancer Awareness Month, you can go to allbloodcancers.org and you will find a link for Blood Cancer Awareness Month. There's a toolkit there that you can use to help us, or help you help us raise awareness of blood cancers and the work that we are doing. There's some stuff that you can do on Facebook. There's an opportunity for you to make a video or send in an image of yourself fighting blood cancers and telling us how and why you're doing that, and other stuff. So go there to our website and click on Blood Cancer Awareness Month to give us a hand. And if you have any ability whatsoever, please donate to the foundation so that we can continue to fund innovative research like we were just talking about, be able to fund education and emotional support programs for patients so that we can, people can be more knowledgeable about their disease, and we can find a cure for this disease so we can put ourselves out of business. So thank you again for tuning in. Again, my name is Carl Alston with the Leukemia Research Foundation, and we will see you again next time.